Dr. Mohammad Asif Khan is an associate professor at the Perdana University School of Data Sciences, Malaysia. He is also a visiting professor at Bismialam Waqif University, Turkey. In conjunction with his induction to the International Fellowship for Outstanding Researchers, programmed by the Scientific and Technological Research Council of Turkey, Dr. Khan obtained his PhD from the National University of Singapore. His research interests are in the area of biological data warehousing and applications of informatics to the study of immune responses, vaccines, viruses, venom, toxins, drug design, and disease biomarkers. He has authored more than 50 international peer-reviewed research and review articles, published in various international journals, books, and conferences, and presented his work at numerous international conferences. He has been involved in the development of several novel bioinformatics methodologies, tools, and specialized databases, and has 3 patents granted. He is currently the elected president of the Asia Pacific Bioinformatics Network. He has done various important roles in the organization of many local and international bioinformatics conferences, meetings, and workshops. Now, I request Dr. Mohammad Asif Khan to address. Sir, please.
All right. Thank you. Thank you for the kind introduction. Can you hear me? Can you give me a sign?
Yes, sir, we can hear you.
Okay, and the screen is visible?
Yes, sir.
Okay, wonderful. Thank you so much. And first of all, I want to thank the organizers for inviting me to give a talk at InSyB2020. It's a pleasure to be here, and I'm very happy to see how InSyB2020 is shaping up. Also representing EpiBioNet, and this is one of our annual events. I thank Prof. Kishore for agreeing to helm this responsibility. Despite not being given enough time, he managed to pull this off, and really happy to see the number of registrants, 900 something. So, and many of them are joining today. And I also want to thank Praj for recommending Prof. Kishore to propose hosting InSyB. Overall, thank you, a big thank you to VFSTR University for all the organizing that you have done wonderfully in order to bring the community together for this year's InSyB. Okay, in the interest of time, I will proceed. Okay, so the title of my talk, as you can see, so I'm not going to repeat, but essentially, this is actually the work of my master's student, Chong Lee Chuen, sure of her later. And you can think of this project as trying to, compress data. It's more of a data compression project. You have used WinZip and others. The only difference is this is for biological sequences. So that's a context that I can provide you at the moment, but let's move forward. Now, this is a picture I think many of you may have seen given the current corona pandemic. And this was a vivid reminder that, corona is not a new pandemic. Well, it's a new pandemic, but pandemics have been part of our history. And we have had a number of them that have been recorded since 165 AD. So we have the Antonian plague that affected 5 million people with a death toll of 5 million people, and many more along the way. A number of these are viruses. We can see that the yellow fever is there with 100 to 150K. HIV/AIDS, which is still a problem. 25 to 35 million people get affected. Spanish flu was a big one in 1918, and that killed, based on estimates, 40 to 50 million, and some estimates go even higher. And we had few smaller ones, but every life matters. So it doesn't matter that they're small or big, but viruses have been part of our history. So with the spread of humanity, well, the threats are also there. Corona is right up there in the front line at the moment. This is an old number, and at the moment, I just quickly did a check on the estimates. We are at the tune of 1.7 million people who have died because of corona. And it's going to be a year soon since when we kind of I think, identified it to be corona. Sorry, yeah, corona around January. So so yeah, this is a sad state, and hopefully through advances in medicine and hopefully championed by bioinformatics in this era, we could do so much more to prepare ourselves for these kind of pandemics. All right, so now viruses are able to do this and become a threat to humanity is because of their ability to change very quickly, especially RNA viruses, as you can see here. Of course, compared to bacteria and eukaryotes, viruses are one of those that change more rapidly, and RNA viruses are right up there. The mutation rate is very high, and of course, they carry much smaller genome size. So, and these mutations, I wouldn't use the word mutation substitutions, because you can have insertion, deletion, mutation, recombination, many things can happen. So the right word would be probably substitution. But of course, in this plot, we are looking at mutation. But in general, so these substitutions, are one of the main reasons that pose a threat because of escape from immune recognition, among other things, which I will cover in a bit. But another thing to remind ourselves is that these RNA viruses, when they infect one person, you could start with a founder virus, starting virus. And within one individual, over time, very quickly, you can have a variant of these viruses within just one individual. Forget about the human population. Within one person, you can start seeing mutations or changes that are different from the parent virus. So, and this population of viruses within that individual are called viral quasi-species. So like quasi, semi-pseudo species. So that's how bad it can be. That within one individual, you can start seeing different viruses over a period of time. All right, so this is one of the main reasons, and one of the good news is that because of the small genome size, we actually have the opportunity to sequence them. Of course, we can sequence things very quickly, and because of their small genome size, there's actually an abundance of genomic data, sequence data available in public repositories. So this is publicly available. Anybody can access these and study them, and this is also what makes viruses favorable and of interest to a lot of people in studying them, given the data abundance. This is just a quick reminder that how fast the data grows. I'm sure we all know that data is growing rapidly, but this is some sort of a data that I asked my student to track, and we track this for a period of 1 year. And for protein, we can see that it has grown from 4.9 million to 5.8 million for a rough period, slightly over than a year. And so that's about 18% growth overall, but nonetheless, close to 1 million new sequences were deposited over that period. And actually, if you look here, this period ends at November 19, and today we are in 2020 and slightly more than November. And I just checked the data again, the number of sequences, we are currently standing at 7.3 million. So this kind of sums up that the data growth is really rapid, and we need to be able to quickly analyze these sequences as fast as possible. And similarly, there's also, of course, a rapid increase in the nucleotide sequence data. I'm not going to go into the details. All right, so given this availability of sequences and the substitutions and changes that viruses go through, I think we need to, we can say in a nutshell that there's a lot of diversity, viral diversity. And I wanted to just quickly break down that diversity into 3 contexts, so that which some of us may not recognize or may not differentiate those when we use the word viral diversity loosely. So there's the genetic diversity. This is simply the diversity at the genetic level, the DNA sequences. Now, so what is protein diversity? Well, not all differences at the genetic level change or translate into differences at the protein level. So that would be your protein diversity. So it's not equal, because you have synonymous changes at the nucleotide level that do not change at the amino acid level. So, therefore, protein diversity would be slightly smaller. In comparison of genetic diversity. Then what is antigenic diversity? Antigenic diversity is more related to the immune system, immune recognition. So all those amino acid changes that you have at the protein diversity level may not necessarily impact or affect immune recognition. especially if you're focusing in the context of certain HLA profiles of certain individuals. So maybe specifically for my HLA profile, not every change would be relevant to my HLA profile. So there is that maybe smaller set of antigenic diversity. So I hope that gives you a little context in terms of the differences between genetic, protein, and antigenic diversity. Now, antigenic diversity is probably, among all the diversity with respect to immune system, this is maybe the most relevant one. I think it's the most relevant one that should be studied when we want to design vaccine or look at therapeutics prophylactic approaches against viruses. Why is that so? Because these diversity, these changes, result in escape from immune recognition. And of course, this directly affects vaccine design. So you could have had a vaccine that has been designed, developed, and if infection happens and the variant is different from the strain that was used for the vaccine design, then there's possibility of escape from that recognition. And this is a problem that we see with influenza. Influenza vaccines go through this annual reformulation so that the vaccine strains, the strains that were used for the design of the vaccine, match with the circulating strains that are going around. And these circulating strains change, so we got to ensure that they match. So there's also this concern about this coronavirus vaccine that have been released. How confident are we that these vaccines would be able to capture the diversity of sequences, of strains that are circulating around? And there was a news about a new UK strain that seems to be spreading quickly. So these are always some of the concerns, and so therefore it's important to study antigenic diversity. There's another thing that we also want to take note of, which is original antigenic sin. This is actually popularly reported for dengue virus. In fact, it was originally observed in influenza virus. So you could read more about it. I don't want to go into details, but the general idea is that you get infected by one and you develop immunity against it, and then when the next infection happens, the next variant is very similar, yet different from the first one. So your memory response gets called instead of a new naive immune response. You call the memory response because of that recognition, but it's not the same. It's slightly different. So what happens is you end up with something called immunopathology. Immune system is trying to get rid of the virus, and it's doing all kind of attacks and all that, if you loosely call attacks, and so you end up with inflammation and other things. So in some sense, you end up damaging the immune system and ends up damaging the host without really effectively clearing the pathogen. So there's this term immunopathology. So this can be a side effect of this immune escape if you have a memory response and you call that memory response, which is not effective. And in fact, it should be calling a naive response because this is different strain from the previous one. So there's a whole different work there that you could read up, but that's another reason why we want to study antigenic diversity and ensure that we try to extract as much as possible from the data, especially when there's a lot of data available. What can we say about antigenic diversity of that particular virus? And hopefully that knowledge gets used when we design vaccine. All right, so in general, study of viral diversity, I think can be easily simply divided into two ways, alignment dependent, and of course the other one would be alignment independent. Alignment dependent is the popular one because it allows us to nicely compare the sequences of these viruses, and we can then, once we align them, we can look at the diversity that's there. So you get conserved, non-conserved regions. The problem with alignment-dependent approach is that it breaks down for very diverse viruses. So I remember we were studying HIV, and there were there's certain positions in HIV that are so diverse. It was really difficult to align those regions, and there were a lot of challenges that we had to go through in order to try to get the best possible alignment that we could. And the other problem with this is it kind of restricts your ability to study diversity beyond a particular species, because the further you go from the species, let's say from species to genus to family, even if the virus is conserved, because you're now studying different sequences under a genus, a species under a genus or under family, the number of species just grow, and therefore to study them together collectively, it's going to become more challenging. Imagine trying to align all the family members. Can be really hard. For some, maybe for some viruses, for some families it's not too bad, but for a lot of families it can be really bad. And what if you wanted to go beyond family? Or what if you just wanted to analyze all known viruses that have been reported to date? That's not going to be possible with an alignment-dependent approach, because you would lose a lot of that conservation. So there's a limit to alignment-dependent approach. Of course, it's useful. We need this kind of approach, but we also need to develop alignment-independent approaches. And there is a lot of, I think, nice work that's going on out there. And so our work here is actually contributing to the pool of tools available to do alignment independent approach analysis. This has been, I think I've seen a lot of nice work for phylogeny with regards to this. But anyway, so I just wanted to highlight that there's this two different strategy when we want to study viral diversity. All right, now, so you could have all the sequences of the viruses that you want to study. You can align them if you're going by the traditional approach. You can't, I mean, you should not study every one of those sequences because not everything might be, especially if you're aligning protein sequences, you don't want to study every one of them because they all may not be relevant to antigenic diversity, as we talked about earlier. Because what might be relevant to the immune system may not be all of those amino acid mutation. Maybe some of them would be relevant. So what would be the best way then to map the antigenic diversity in your sequence that you are interested in studying? Of course, wet lab would be the best way to do this. To study and determine which one of those parts of the sequence are epitopes. And we know from papers that not many of those regions would be epitope. Typically from 1 to 5% is thought to be epitope. So you're going to spend a lot of money, a lot of time in trying to study all these sequences. It's just not effective because the number of tests that you would have to carry out is just enormous. And this is just for maybe one species. Imagine if you wanted to study all the viruses out there, it's just not possible. All right, we would need something more high throughput in order to deal with the big data. Even then, it's I think a computational approach is necessary before we go to wet lab. So there's wet lab would be the best, but there's a big limitation there. Of course, the next one would be in terms of computational approach. We could predict the epitopes. Could we predict the epitopes? But unfortunately, we don't have prediction models for the different repertoire of people's immune system. For example, my HLA profile might be different from others, and the last I checked, there are like 25,000 different actually molecules that have been reported in the for the human population, and that's just what we know. There might be so much more out there. So and we don't have a prediction system for each one of those alleles. Of course, we have the concept of HLA supertype and other things, and we could look at it from that context. But nonetheless, the point here is there's some limitation if you wanted to just do prediction. And of course, some of those predictions might be wrong. So this could be an overestimate, but this is still better than doing a wet lab. So we wanted to use the computational approach, but we couldn't do the prediction approach because of that limitation in terms of computational models available. So then we actually decided to kind of treat the whole sequence as relevant for antigenic diversity, and we made this on two bases. We made this assumption on two bases, that if we are designing vaccine and we want to consider the whole human population, and of course this is in the context of cellular immune response. If you want to consider the whole human population, then we have 25,000 plus HLA molecules, and for a given HLA molecule in a protein, you would have at least 1 to 5% that would be recognized. So if you now consider all the HLA molecules, maybe 100% of the protein would be relevant. If we are looking at the human population level, at individual level, of course, it's going to be much lower, but at human population level, it would probably come pretty close to the whole sequence. So the whole sequence is relevant to somebody in terms of maybe recognition by the HLA molecules of the immune system. So in that context, then we assumed that everything would be relevant for antigenic diversity if we posited at the population level. So therefore, we thought, given that, then we don't need to predict the epitopes. We just study the whole thing, and of course we use a k-mer of a certain length because epitopes are of a certain fixed size in general. So the size that we chose is 9 amino acid. That's the typical size of HLA class 1 epitopes, and it's the core of HLA class 2 epitopes. So we decided to use that size when we study the sequences, and we can use a sliding window, so 1 to 9, 2 to 10, and then we look at the diversity within those k-mers. So moving on, the idea of a minimal set. So I just wanted to quickly explain this. So we talked about studying the whole sequences. So we take a sequence and we study the whole thing in terms of, for our antigenic diversity analysis. So here's an alignment just to illustrate the idea of a minimal set. Of course, what we did eventually is not non-alignment based, but this really helps understand what do we mean by a minimal set. So imagine you collected a bunch of sequences and you remove duplicates. So it's a given that you remove duplicates. So you end up with 3 sequences, and these 3 sequences are unique. As you can see here, there's F. They are the same, but there's an L here, and there's a Q here, so that makes this sequence unique. This sequence was already unique from the other 2, but there's also Q, which makes it unique. So the Q's match here, but the L is different. So the second sequence. Well, essentially, you can see from here that all the 3 sequences as a whole are unique from each other. So now what you do next is you generate the so-called k-mer. We chose 9. I've already explained why 9. You could do different k-mer length also. So you create this repertoire of overlapping 9-mers that you can get from these sequences. And this is what we could refer to as the peptidome, or the viral peptidome, or the peptidome of our data set. So you generate this peptidome of a certain k-mer size from each one of those. So these are the k-mers. And then you try to see what is the minimum number of sequences you would need to capture all these k-mers. So you can see from here that all the k-mers here are shared among these 2 sequences, and all the k-mers here are shared among these 2 sequences. And these k-mers are shared across all the 3 of them. So we could immediately see that actually one sequence seems to be redundant in terms of k-mers. So you could just use 2 sequences and capture all the repertoire of, sorry, you could just use 2 sequences to capture all the repertoire of k-mers that were there in the data sets. All right, so this sequence is the redundant one, and you could just use these 2. So 2 out of 3, so that's the reduction you make. You can lose 1 sequence and you wouldn't lose the repertoire. All right, so that's the idea of the minimal set, and this is actually something that we had applied previously about 15 years ago. This was a work that I did under the supervision of Prof. Bruzik, and we wanted to revisit this because this work was applied on dengue, was applied on influenza, but we had not applied this beyond those 2 viruses, and it was quite interesting to us to see how this would actually pan out for a lot of the other viruses out there, especially if we wanted to analyze all viruses known to date, the sequences of all viruses known to date. So we set out mapping the minimal set of the viral peptidome. And by viral peptidome here, I'm referring to all known viral sequences at that point when we did this study. So to break down the objective, we wanted to look back at the algorithm, and the algorithm, we couldn't find the file, it's been that many years. And we had to kind of build from scratch again. And we also realized there were a lot of issues with the algorithm, not in terms of being big data scalable, not in terms of what it could do, but in terms of big data scalability. And it was also not optimal in many ways. So we ended up trying to optimize it and make it big data ready first before we went to do our analysis. And then we also wanted to understand what are the factors that can affect the minimal set of sequences, and then we went out to generate the minimal set. And then I'll briefly talk about how we applied. We went on to investigate how this affected across lineages, viral lineages. So this is a quick methodology overview. So you collect the data. Of course, we went to NCBI, we collected the nucleotide and the protein data. We put the nucleotide aside just for reference, and we went on with the protein data. We removed the duplicates, and then we optimized the tool, and then we investigated the factors, and eventually we did the analysis. And I will go through this. I'm going to skip this. All right, so this is the data collection part. We can skip this. We just explained. All right, so let's focus on the optimization of the algorithm. So this is actually the old algorithm. This was published as part of my thesis then, and I'm not going to go into the details, but essentially the idea is that you have all these sequences, you generate the peptidome, you do this overlapping k-mers, and then you have this k-mers, and of course you remove duplicates, you end up with unique k-mers, and then try to see how many sequences you need to capture all these unique k-mers. So, so first sequence might capture all the 50% of them. With the second one, cumulatively you capture 91, and with the third one you're already capturing 100%. So you only need 3 out of all the many in order to capture these. All right, so we ended up developing 3 algorithms: iter mean, rank mean, and unique mean, because we were just trying to progressively explore how to make it better. So the first one was was accurate, but speed-wise was very slow. So it was not big data ready. And we're talking about like 5 million sequences. So we definitely needed speed. And one estimate gave us that we would need 7 months with iter mean if we didn't optimize the speed. So we just couldn't wait for 7 months to see the results. So rank mean, was speedy, but it suffered in terms of accuracy. And then we eventually came up with unique mean that was accurate and also very speedy. These are just some of the limitations of the old algorithm. I'm not going to go through this, but as I mentioned, there were a number of limitations and we wanted to overcome those, and eventually unique mean in some sense overcame all those limitations, nearly all of those limitations. All right, so I'm going to skip that. So here are some results just to give you a flavor of how they performed. So with 1,000 sequences, that was actually the actual data that was used for Ka-Khan algorithm back then, compared with the new sequences that we use for the other tools. So we don't have the data for this because this was not done back then. But in terms of reduction, there was a reduction of 9.4%, but iter mean, as you can see, was very slow. But was able to reduce at 26, so it was much better in terms of reduction. So there's some optimization that was done, and we could actually achieve much more compression. And rank mean didn't perform very well in terms of the reduction, but actually was quite fast. And unique mean was equally fast, but reduction matched iter mean. So accurate and fast. And we wanted to be sure whether this less than 1 minute was always going to be something like that or close to that. What if the data was quite big? Would we still see a very speedy analysis? Of course, it would still be fast, but how fast would it be? Just to get a sense of that, we actually then tried it with 10,000 sequences. As you can see, iter mean was very slow, and we got a compression of 43%, and rank mean was fast, but there was a problem there. Unique mean was able to do this in 8 minutes, so that was still acceptable. 10,000 sequences, 8 minutes. Of course, with 5 million, you'll probably need about 2 months, which we could, tolerate, but actually ended up being much less. If I'm not wrong, about 18 days, we were able to get the results for the 5 million. Anyway, so this looked good, and we felt that we were ready to then proceed with our next step. So in conclusion, unique mean offers the best combination of speed and optimality to generate the minimal set. And we were able to actually achieve this thing with unique mean because we recognize there's something called singleton monomers that are only found in one sequence. So immediately they become candidates of the minimal set. So what we did was we tried to filter them out, and that really reduced our data set, and then we could do a lot of things much faster. So there was this thing that we recognized, and we separated, and that actually speeded up things. And of course, there was some other tweaking that was done, and some other codes that we used. And of course, multi-core, being able to use multi-core was one of the other reasons for that performance. And with better compute, this should perform much better. And of course, so we eventually, and we have in some sense demonstrated that the tool is applicable to big data. All right, moving on. So we then went on to analyze the factors affecting the minimal set. So these are the list of factors that could affect the generation of the minimal set. The sliding window size, what k-mer you use, and I explain why we need to study in the context of k-mers. Sequence number, how many sequences. So big data, of course, would affect the length of the sequences. Proteins are of different length. Viruses, proteins can be of very different lengths. And the region, like in terms of conservation, if you're studying only conserved sequences, diverse sequences, of course, they would impact the minimal set in a different way. And actually, these three were already studied in the earlier study, except that for the sequence number, the number that was studied was much smaller, and we wanted to revisit that with a much larger data set. The other thing, of course, that was not studied is sliding window, so we wanted to also investigate that. So this time round, we focused on sliding window and sequence number. But what did Khan et al. Observe for sequence length? For sequence length and sequence number, well, in general, if your sequence number increases, you can, well, intuitively you are not able to compress much. Okay, your ability to compress reduces with growing number of sequences. And in terms of sequence length, with growing number, with larger proteins, of course, it was much harder to compress because of larger k-mers that you would generate, and sequence region also, it varied. For example, highly diverse regions would be harder to compress compared to highly conserved. So it was all intuitive, but it would be good to capture these in terms of the actual plots. So then we set out to do that, and of course we wanted to make sure that we are correcting for confounders. So when we want to study the k-mer, confounder would be the diversity of the sequences, the number of sequences, the length of the sequences. So we needed to control this. And for diversity, we used Shannon entropy to study that. And this is a formula for Shannon entropy. I'm not going to go into details. You can read our papers. And we have actually applied this on a number of viruses. And the highest we have seen is actually 10 observed for HIV, clade B. Theoretical maximum is 39 that you can get for monomers. And the highest we have observed is 10. So 10 is, so HIV is rather plastic in that it is able to achieve that kind of entropy level. And entropy, basically, the higher the value, the more diverse is the region, the lower the value, the more conserved is the region. So we actually then, given the data sets that we wanted to use, we thought we needed to classify the diversity into 4 areas: conserved, semi-conserved, diverse, and extremely diverse. And based on our understanding of what we have done in the past with previous viruses, we were able to identify those that would fit in each one of these bins. So we further classified this part 1 here into 4 bins so that we could understand the minimal set with respect to each one of these levels of conservation. And so these were the data sets that we used to do our factor analysis. You can see the entropy value would correlate with the grouping that I mentioned earlier: conserved, semi-conserved, diverse, extremely diverse. And for the sliding, when we did the sliding window size, we explored 3 mers to 23 mers. This is the range that we felt was relevant for immune system. And not just, of course, for cellular, it doesn't go to 3, but I think in terms of antibody epitopes, some of them can be much smaller, but we just, for curiosity's sake, we just went from 3, which is where I think most people say is non-random, begins to be non-random. BLAST uses that 3 amino acids. So we went from 3 to maximum about 23. That's the range I think class 2 can recognize. So we took that whole range, and when we did that, we fixed the number of sequences and we fixed the length of sequences. So number was fixed to 100, the length was fixed to 100, and then we of course went diversity by diversity, different diversity levels, and we went across the different k-mers. For the number of sequences, we analyzed from 100 to 1000. Again, this is not really big data, but it was much larger than what we had done, Khan et al. Had done previously. And of course, when you do this, you fix the length and you fix the window size, and we chose 9, and which the reason I've explained before. So this is the result now, factor sliding window size. So in general, what we could say is that the reduction, antigenic reduction, basically how much could you compress? So the antigenic reduction was the highest for the conserved, and I think that's intuitive. Conserved, there's a lot of k-mers that would be redundant, so you could really compress them. And then semi-conserved next, diverse next, and extremely diverse was the lowest in terms of compression. But we also noticed that, as the overlapping k-mer size increased, the ability to compress also reduced. It was more or less flat for conserved. It didn't change much. Similarly for semi-conserved, for diverse, it started also flattening out at around 23. And similarly for extremely diverse. So there was this, you were able to reduce further, and then it just started to flat out. All right, so that was the observation for sliding window. And in terms of sequence number, as I mentioned earlier, Khan et al. Had studied this, and if you increase your sequences, your ability to compress just reduces. But nonetheless, of course, you can compress, conserve and semi-conserve a lot, followed by diverse and extremely diverse at a small number of sequences. But as the number of sequences increase, your diversity, well, the pool of variation, because these are unique sequences, right? So there would be natural diversity increment there. So then it starts tapering off, starts flattening out in terms of reduction for the semi-conserve and diverse. And we actually were quite curious to see how this would actually go for the extremely diverse, and we observed later on. So this was a unique observation that extremely diverse actually can continue to go up to the point that we have seen in some cases it actually beats the compression you achieve in terms of percentage with the conserve and semi-conserve. Right, so there's a bit of you should actually redo this with much larger data set, and there's a twist here that extremely diverse could actually overtake diverse and semi-conserve in terms of how much you can compress. All right, so we will, bearing that in mind, proceed. So in conclusion, for this part, antigenic reduction was inversely correlated with increase in k-mer size. So you increase the k-mer size, your AR reduction, the ability to reduce slowed down, you flattened out. Antigenic reduction was directly correlated with increase in number of sequences. So as you increase the number, your antigenic reduction also increased. And AR was the highest for the conserve and the lowest for extremely diverse data set. But as I mentioned earlier, this actually flips for some viruses with a lot of data and a lot of diversity. So moving forward, generation of the minimal set for all known viral pair sequences. So this is when we really set out to do what we wanted to do from the beginning and apply this on all viral sequences, all those close to 5 million back then. So we took all our sequences. First, of course, we removed the duplicates. So it was nice. It was interesting to see that half of the sequences were duplicates. So that's an observation. And then we were left with 2.1 million, and we were able to compress this further, not so much actually, only about to the tune of 16%. That was the antigenic reduction through unique names. So we ended up with 1.3 million sequences. And now as a whole, I think if you sum up together the duplicate reduction and the antigenic reduction, that's a total reduction of 73.4%, which means we are only left with 26.6% sequences of the input sequences that would be relevant in terms of antigenic diversity. So that's a huge reduction, and there are a lot of implications of this, which is that if you wanted to study antigenicity and all that, you could be just focusing on this 1.3 million sequences, especially for those people who don't have a lot of compute. This would be much more manageable data size. And also if you wanted to do wet lab experiments, this is definitely much more manageable number of sequences to be studied. Well, still big, but much smaller than 4.9 million sequences. So there was this interesting observation that how much we could reduce in terms of totality, but antigenic reduction itself was actually not so much. So the 56% reduction, the high reduction up there implies that there's a lot of maybe sampling bias, there's a lot of duplication that's going out there. And the antigenic reduction being rather low indicates that there is still a lot of inherent diversity within the sequences, and therefore that we were not able to compress much there. So in conclusion for this one, the high duplicate reduction reflects possible sampling bias in the collection and reporting of viral sequences in public databases, and this is there, we have seen it, usually sequencing would be not spread out. People would be going for the same sample sites and maybe neighboring sample sites. So there's that tendency to pick sequences from a certain sample sites, rather than be more widespread, more uniform. And relatively low AR observed for the viral peptidome suggested a high diversity of the viral superkingdom. So the viral superkingdom, it's actually very diverse in general. And we can, of course, we can relate to that with the fact that there's so many different species of viruses out there. And the AR we predicted that this AR will range from 15% to 25% with over time with further increase in data. The reason for that is you already have all this diversity captured. New sequences that are coming in are not going to drastically increase that diversity. I mean, in terms of, let's say, k-mers, you're not going to get a lot of new k-mers. Even if a new sequence comes, majority of the k-mers will be exactly the same as the previous sequence. Maybe there's one mutation here and there. And those mutations will result in some new k-mers, but the majority of the k-mers are going to be more or less the same with the existing data. So in terms of the AR compression, we think that this is, well, 5 million is quite a big data. So we think that this number is not going to change much. Will go up and down. Why would it go up? Because, well, if this existing data is not really representative, we end up sequencing some novel viruses, then there would be some changes. But if you're not going to sequence something novel out there, and this is reflective of what's out there, then of course it's not going to change so much. But nonetheless, this is what we think it would be, and of course it'll be interesting to do this maybe few years later, just to see what's the status then. And the last part, actually, I'm not going to go into detail because of a time issue. But once we had built the tool, we applied it on all viruses, and we saw what it looked like. Actually, the next thing we wanted to do is to do this across species, genus, family. So like take one sequence, take one species, and then take the genus of that species, then take the family of that species, and try to chart out this landscape of minimal sets across this lineage. And so we have actually a lot of results there that I don't have the time to go through, and hopefully that will be another talk someday, and we'll be able to share. But also all this work is currently being prepared for publication, so you hopefully will be able to read these results in the near future. So that's really one nice application. Now, as I mentioned, the issues with alignment-based approach, you could mostly do it at the species level. Going beyond, it's going to be very challenging. So with this tool, we can now study the diversity at a species or a genus, at a family. We could even go where we have already gone to the root, which is all the super kingdom, which is all viruses, and we've already shown the results. But we wanted to now zoom into the various lineages and see how it changed across lineages. All right, so in the interest of time, I'm not able to go into those results. I'm just going to conclude now. I think I'm more or less hitting the time limit. So our tool, Unique Mean, which is publicly available, enables analysis of a minimal set for a given sequence data set of interest. And as I mentioned earlier, this is really an enabler because it allows us then to go beyond a particular species, to go beyond alignment, and to be able to study any viral sequence that we are interested in. And of course, actually, the tool would work with other organisms, for example, bacteria, if you wanted to. We have not tried this, but we think it would also work with bacteria and others. Actually, we had a project to do the same thing with bacteria, but bacteria, we have so much more data, 700 million sequences. So for that, we really needed much more compute power. So that's something ongoing, and hopefully we'll have some results in the future for that. The project is a little on hold because of that compute, and hopefully we'll be able to share something there. So the tool is an enabler. I'm not saying it's perfect tool. It probably needs more improvements, and hopefully we can improve this and make it much more robust in the years to come. Also, I think the second key point is Unique Mean is able to reduce big data to a reasonable working size, and there's a lot of value in that in terms of kind of zooming in and helping you just focus on the sequences that you should be focusing on when you want to study antigenic diversity. I think it has some positive implications to wet lab validation. You don't have to be overwhelmed with all the data that's out there. You could focus on much smaller data without worrying about losing the possible repertoire of k-mers that would be immunologically relevant. And also this is very useful for those people who don't have the compute. Not all of us are blessed with a lot of compute power to do this kind of analysis. So this can also be helpful in terms of shrinking the data size so that you could focus on a much more manageable data set. And lastly, the low AR antigenic reduction that we saw when we did all viruses, observed for all the viral peptidome, suggested that the viral superkingdom is actually inherently quite diverse. If this is what we have seen, then it's going to hover around that. But if this is not everything that we have seen, and there might be a lot of novel viruses that are yet to be sequenced, which I believe that would actually more likely be the case. I don't think we have sequenced everything, so there would be some novel. For example, Mimi viruses was a novel discovery a few years ago. Giant viruses. I'm sure that if we do these fishing expeditions, we might discover a lot more novel viruses. So, but nonetheless. We estimate that this is going to range, the AR is going to range from 15% to 25%. So you at least get some rough idea what kind of reduction you can get. But for the DR, duplicate reduction, it's very hard to say what we will get because it really depends on the sampling and it varies from virus to virus. So that's something, it's a bit hard. For example, you could get a duplicate reduction of 90%. You just reduce by 90%. And this we observe with Ebola virus. And for some viruses, maybe you get much lesser. So actually, this is something else we wanted to go back and look, do a factor analysis on duplicate reduction and see what we can learn from there. So that's all I got to share with all of you. This is the GitHub page of the student Chong that was made by the student Ms. Chong, and the tools are available. So if you're interested in applying this, we're happy to collaborate and work with you. And I just want to end with acknowledgement. Again, a big thank you to Chong, whose photo is here. She did all the hard work here, and credit goes to her and bringing the project to fruition. And she has completed her master's, and I want to acknowledge Dr. Kenneth Ban of National University of Singapore for giving us some compute power to run these to run the tool. Dr. Choi Sui Bing, my colleague from Perdana University, for assisting and co-supervising the project, and Mr. Lim Wei Lun Liner for working with Chong and doing some of those code tweakings in optimizing the code. And last but not least, Vladimir Bruzik, my former PhD supervisor, who kind of initiated this work about 15 years ago. And we also received these fundings to support the project. Thank you very much. And I'm happy to take any questions. I hope I didn't exceed the time. I have kind of lost in terms of time. So I hope I'm still okay, or my apologies if I exceeded.
All right, sir. That was really a great talk. And now I would like to ask the participants whether you have any questions. We are now open for questions for 1 or 2 minutes.
Akila, shall I ask now?
Yes, sir. Please go ahead.
So, Dr. Asif Khan, sir, it is a wonderful talk on viral peptidome and antigenic reduction. So, in this, there is 1 or 2 queries are there which I would like to get clarified from you, sir, Asif Khan.
Sure.
So, what nowadays mRNA vaccines are coming, will they be promising, or do they replace our antigenic vaccines? If they replace, what is the procedure, how to trap mRNAs in a large scale? What we are doing with the protein collecting, trapping in a large scale is convenient rather than mRNAs collecting and trapping in a large scale, ample.
Right. Thank you for that question. This is definitely very relevant and pertinent with mRNA vaccine approaches being used, to combat corona. So in terms of the antigenic diversity, I think at the end of the day, whether you use mRNA or DNA, that's more of a delivery mechanism. At the end of the day, the immune system recognizes peptides. So the actual recognition happens at the peptide level. mRNA, DNA vaccine approaches, which were there before that, facilitate the delivery because peptides are not so stable. So when you infect with a peptide, they may get degenerated easily, and there are a number of issues with that. So I think these approaches are taking care of the delivery mechanism. Eventually, what the immune system recognizes. I have not read much of this yet, so I might be off, but what I know from the immune system is that it will recognize a peptide. So at the end of the day, you still need to deal with peptides, protein sequences, and the mRNA is going to code for a protein, which will hopefully will get chopped accordingly and that be presented to the immune system. So this is still relevant, and this is still, it kind of doesn't change in terms of actual recognition by the immune system. Even antibodies, I don't think antibodies are going to recognize the mRNA, but rather it's the protein that's coming up. I think this is still relevant.
With regard to the delivery mechanism, there must be some, what is it called, techniques that we have to develop, particularly for mRNAs.
Yes, and I think this is a novel area, and I see a lot of promise there.
Yeah.
And
another important feature, Asif Khan sir, another important feature, you were mentioning about the conserved regions and antigenic reduction, all those things. What I wish to let you know is, if there are conserved regions among 5 or 6 pathogens, if we prepare a vaccine taking into consideration the conserved regions, will not this conserved region will fight against those 5 or 6 pathogens for which we designed the vaccine?
Yes. Actually, this is the premise behind the search for universal vaccine against influenza. Influenza, for example. So
H1N1.
Yes, influenza. So there are a lot of papers that talk about universal vaccine, and the idea is if you find something conserved that's across the board, then it would work. The problem is with influenza. Actually, my colleague Heini, a long time ago, for 13 years ago, actually published this paper on H5N1. When you go look for something that's conserved across different subgroups of the virus, for example, different subtypes of influenza, you do see something that's conserved, but the number of these sequences that are conserved across is much smaller. You don't have that many of them, and even if you do, they're very short. So then you don't have a lot of sequences to play with in terms of trying to see if they're really optimal in terms of immune recognition. So that's one challenge out there. Of course, the alternative, and in dengue also we did an analysis and we proposed something called pan-dengue virus sequences, sequences that are conserved across each one of the dengue. And the idea was that this might mitigate the original antigenic sin because it's conserved across the 4. And we found 44 sequences that were conserved across the 4, but they were not 100% conserved. So there were still some small variants within each of the serotypes. But we found 44 that were shared at 80% or more conservation. And there were like 6 only that were 100% conserved. So your number shrinks when you're looking at higher conservation. And if these 6 turn out to be not immunogenic or poorly immunogenic, then you're left with nothing. So that's the challenge with the universal vaccine, but definitely that's the idea if we could. So the alternative to this so-called pan or universal vaccine is to avoid original antigenic sin, and we have just published a paper on this, which is to look for like species specific and highly conserved. So you could find something that's maybe in the case of influenza, let's say H5N1, highly conserved in H5N1 and species specific to H5N1, and then find something that's highly conserved in H7N9 and specific to H7N9. So there's no cross-reactivity, because cross-reactivity is something that we don't fully understand, and this is where original antigenic sin can happen. So for dengue, we proposed, you find like serotype 1 very conserved, but different from serotype 2, and different from serotype 3, different from serotype 4. So when you get recognition, immunity, memory response to that, this memory cell will not get confused in recognizing the other serotypes, and for the other serotypes, you will call the other response. So there's a lot of work going on in this, and both ideas are interesting, and hopefully something comes out of that.
Another interest, conserved regions need not be RBD, receptor binding domains.
Yes, it did not be. For example, in the case of cellular immune response, the protein gets chopped, and then and so you have the availability of these chopped peptides that will then bind to the HLA. And the HLA doesn't prefer something that's from the RBD. It could be any part of that protein. So for cellular immune response, it's RBD. It doesn't matter.
So one more important point, Khan sir. Your Malaysian people HLA and Indian people HLA, if you make the vaccine, will it differ?
Well, in Malaysia, we do have Indians there, so maybe there will be some commonality there. But you also have the Malay population, you also have the Chinese population, and there will be diversity given that. And also in India, you have a huge diversity in India itself. I mean, India is so big and so diverse, so so you would actually struggle to find commonality among Indians itself, if you consider the whole population.
Sir,
But
the last point is the SARS-CoV-2. If you prepare a vaccine, will it work against the MERS and the SARS-CoV-1 also?
Okay, that's a very good question, and I was very surprised to see that the MERS sequences are actually very different from the current SARS. If I remember correctly, they were like only about 20 to 30% similar. So there's a lot of diversity. So most likely they won't work because these coronaviruses are quite different from each other.
Yeah. Thank you, sir. Whenever you come to India, please tell us. We'll host you for some time.
Thank you. My pleasure. I was happy to be part of this. Thank you.
Yeah, we've got good friends from Malaysia. They came to our place and spent here for a week time.
Nice, and likewise, if you come to Malaysia, do let us know.
Yeah. So with the permission of the chair, I just have a small question to Asif. Yeah, please. Yeah. Yeah, Asif, it's a wonderful. It's a wonderful talk, Asif. Thank you so much. So I was just wondering, how minimal is a minimal set? Can we just draw to a conclusion that they must be, or they could be, this much minimal? Just like in your case, you have drawn to a conclusion that you have reduced to 44%. But what if, if a future pandemic, would probably come up with a new viral peptidome data sets? What would be the minimal for a minimal data set?
Well, the minimal for the minimal data set should be whatever is in nature. I think nature would know the answer to that, and we looked at the available data that has been reported from nature, and what we got was for all viruses, the minimal data set was 26%. So you could compress it by 73%, and you're only left with 26% that would represent. So that's what nature told us based on what we have. And as we see more of these viruses, and if we discover novel viruses, then we will get closer and closer to the truth, the actual truth number. So, I mean, overall nature, we don't have all the data. So that would be the truth. And what we observe is, a reflection of that truth. So we are right now standing at 26%. And with more sequencing, we will get closer and closer. And we suspect the actual number, as I said, hovering between 15 to 25%. So the final, let's say if we could sequence everything available in nature, then we will know the actual truth value, but we are not there. But we suspect that actual truth value would be somewhere between 15% to 25%. So we don't have any particular desire that it should be here or it should be there. It's really what it is that nature would elucidate to us eventually as we sequence more of these. But we'll probably never get there. Yeah.
Thank you for an excellent talk. Yeah.
Thank you, Prash.
Thank you very much, sir.